Muito boa tarde a todos, sejam bem-vindos a esta casa, que é de todos nós, e portanto hoje vamos ter o privilégio de estar com estes nossos conferencistas que vão enriquecer não só a nós que aqui estamos presencialmente, mas também aqueles que nos seguem ou pelo menos estão connosco via Zoom e aqueles que mais tarde nos acompanham seguindo a conferência no YouTube. De maneira que, muito obrigada por terem vindo e é para nós um prazer e espero que para lá do privilégio de termos estas conferências notórias hoje, terão também, seguramente, o privilégio de passar, pelo menos, esperemos que tenhamos o tempo certinho e de terem, no mínimo, ou no máximo um quarto de hora para sair por outro lado e verem ou passarem um pouco à vol do azul, como se costuma dizer, pela academia e terem ou criarem a curiosidade de voltar, não só para conferências, mas também para visitar esta casa, que é um tesouro, e é um tesouro que é nosso. Let me now introduce or compliment our guest and our professor, Philippe Mancia. I have to thank you very much for coming here and to be with us, because it's a privilege to have you as our speaker in this meeting. So thank you for coming, and I hope this is the first time maybe you come to the Academy of Sciences, but we may have in the future many other opportunities to have you with us. For the moment, I would like only to give you a very, very warm thank for being with us, and Lena will charge of introducing all of you, all the speakers that we have today. Other than is Ana Messias, João Cabral, and Pedro Matias, and for you also, I have to thank to be with us, talking with on a so important subject, the celebration of structural biology. So thank you all, and please think that your contribution is very, very important for the life and scientific life of the Academy, and your contribution will be not only today, but all along the time you want to be with us, very important for the scientific community in Portugal, and not only in Portugal, but around those that can understand the Portuguese and the English that will be translated in also in the YouTube. Thank you for coming, and have a good day.
Thank you very much, Professor Maria Salomé Paes. Well, I'm delighted today with the fortune of being able to celebrate prominent developments in the area of structural biology in this magnificent library, in this historical institution, and since 1 year ago, I dare considering as home. I truly thank the President of the Instituto de Altos Estudos, Professor Maria Salomé Paes, for embracing without hesitation the idea of organizing this symposium under the umbrella of this prestigious institute. Last, I want to address a warm thank to Dr. Nuno Borges and Dr. Ana Esteves, my ex-collaborators but forever friends, for their invaluable contribution to the organization of this event. To be honest, my initial idea was not so much celebrating structural biology, but celebrating the 10-year-long wonderful and fruitful cooperation between my lab and the lab of Professor Philippe Mancia from Columbia University in New York. One evening, I received a call from someone who I had never met to tell me that he had a problem related with hyperthermophiles, and I was the only person in the world who could solve it. Philippe has the rare ability of being very enthusiastic, positive, and motivating everyone around him. Unfortunately, he was wrong. We tried hard, but we could not provide a solution. However, since that first contact, we engage in a nice and fruitful collaboration that lasts until today. So this is primarily what we wish to celebrate: 10 years of great ideas, great learning, and rewarding social contact with Filippo and his team. Thank you again, Filippo, for behaving as a wonderful catalyst to make our work advance faster despite the ocean that separates us. And now a very special and sincere acknowledgement to the other three speakers who generously accepted our invitation to contribute to the success of this hearty celebration. And we'll start with Pedro Matias, that I call now. I will say just a few words because you can read the bio if you want. But Pedro graduated in chemical engineering in Lisbon, obtained a PhD in crystallography from the University of Pittsburgh in the USA, spent a postdoctoral period at University of California in Berkeley, and finally, in 1989, he returned to Portugal to join the protein crystallography group at ITQB. Pedro is a top protein structural biologist, and I'm sure that most of the success of ITQB in this area was due to his merit. Pedro, thank you for accepting the challenge of telling us about the journey, the 7-year-long journey of protein structural biology. Some technical problem?
We should fix it.
So, first of all, I would like to thank Professor Helena Santos for inviting me for this presentation. This is a bit unusual because it's not purely scientific, but it does contain some elements of science, and I hope you will bear with me and do not mind my mentioning a few equations which are fundamental for our use in structural biology. But I have to start this presentation at the very beginning, not the creation of the universe, but the discovery of X-rays. I need to hide this. So, X-rays were discovered by a German scientist called Wilhelm Conrad Röntgen in 1895. And at the time, they were called X-rays because nobody really knew what they were. Although the Germans have always called them Röntgen rays, all for obvious reasons, because they were discovered by a German scientist. And not long after, he took the first radiograph, which was an image of the hand of his wife. And it was also a bit puzzling and even downright mysterious that you could have a radiation that would go through the soft tissues of the human body like they were not there. And then on the other hand, you had the contrast that was provided by the more dense bone material. And so, of course, as a result of this, Röntgen is credited with the invention of radiography, which is a modern, which is used as a diagnostic tool to this day. Later, another German scientist, Max von Laue, they had an idea. Okay, so what happens if we shine an X-ray beam on a crystal? So, of course, crystals were known at the time, and I think this was copper sulfate that he used. And you can see that that picture over there, it's the first X-ray diffraction image. Doesn't look like much, to be honest. but it was a surprise because you could see discrete spots in the picture. So immediately, the question arose, why do we see those spots? But first, we had to try to understand why can we produce X-rays, or where do they come from? The interpretation actually was not possible until the early 20th century with the development of quantum physics. But already in 1913, X-ray tubes were available, like this one that you see here, where you shine an electron beam on a metal target, and then you produce X-rays coming out of the target. And the energy of the beam depends on the chemical nature of the target. So when you increase the voltage applied in the X-ray tube, which also translates into kinetic energy that is given to the electron beam, you start by seeing first, you don't see pretty much. After a certain threshold value, you start seeing strong discrete spikes on the emission spectrum of the metal that you are using as a target. In this case, this is for copper, which is a metal target that is commonly used for protein crystallography. And you have these two lines here, K alpha and K beta, which are overlaid onto a continuum of radiation. So why does this happen? It happens because the energy of the electrons is high enough that it can eject an electron from an inner core orbital of the copper atoms, the 1s electron. And because then there is a hole in this at this energy level, there is an electronic transition from a higher energy orbital down to ground state. And then as a result, you have an emission of a photon with X-rays with energy corresponding to the difference in orbital energies. And so this is where the K alpha and K beta lines come from. For other elements, you start having involving the d orbitals, and then you start having L lines and not just K. So, okay, now we know about X-rays, we know where they come from, how they are generated. Why do we see diffraction? How can we see diffraction? So diffraction comes from the very interesting fact that the wavelength of the X-rays has dimensions which are similar to the size of an atom. Even though the atom, you cannot really assign a definite size to an atom, there's an approximate size that you can consider, which is the atomic radius. And they are all in the ballpark of 1 angstrom, 1-point-some angstrom length. And as a result, when you shine an X-ray beam on an atom, it is scattered in all directions by the electron cloud around the nucleus, and scattered regions or scattered beams from different regions of the atom will interfere with one another, and they will begin to cause destructive interference as a function of scattering angle. So just to refresh your memory on this, when you have constructive interference, the waves are in phase and reinforce each other. When you have destructive interference, the waves are in opposition of phase and they completely cancel each other. Of course, here you have something in between. You never really get a fully destructive interference because we're only dealing with one atom. So now, but what we see is not scattering, although we can do that experimentally. It's not X-ray scattering by atoms, but by crystals. And crystals introduce another element in the equation, which is three-dimensional long-range order. So crystals are built by the repetition in 3 dimensions of a fundamental building block, which is called a unit cell. And as a result of that periodicity, then the pattern that we see in terms of diffraction is made up of discrete beams and not a continuous function. You should be able to see here something. Okay, it doesn't play in this computer as well, a video. But anyway, this is a diffraction pattern. and you can, as the crystal rotates, you can see the diffraction beams coming in and out as they go out, coming in and out of diffraction position. So the arrangement of these diffracted beams can be described by a mathematical construction called reciprocal lattice, which is related to the crystal lattice. And like the crystal lattice, you can also define a unit cell for the reciprocal lattice, and you define also edges and angles similarly to the crystal lattice. The difference is that whereas an atom in a crystal lattice can occupy any position, so the coordinates are given by 3 real numbers, x, y, and z. In the reciprocal lattice, you can only have discrete beams, so the coordinates are given by 3 integer numbers. And this transition from continuous function to discrete diffracted beams is imposed by the translational symmetry of the crystal lattice. So then this was interpreted as arising from reflections from a family of parallel planes with a certain interplanar spacing. And then it was possible to see or to deduce that the constructive interference could only arise when the reflected waves were in phase. So in this case of the simple diagram, so you have the difference in path length has to be an integral number of wavelengths, and this is what is known as Bragg's law. So Bragg's law is actually, there were actually 2 Braggs, father and son, and as a result of this discovery, they earned the Physics Nobel Prize in 1915. But we can now say that after, I'm sorry to have to expose you to these mathematical equations. You don't have to learn them, but it's important to explain what's underlying them. So when you have a crystal structure, we can define a quantity called the structure factor, which is a function of all the atoms in the crystal structure. And the equation is given like this, where you have then the structure factor is a complex number, which is composed of an amplitude and a phase angle. By the same token, you can, once you know the amplitudes and the phase angles, you can calculate back the electron density at any point in the crystal structure by using this other formula, and which is a Fourier transform of the structure factors. So both electron density and structure factors are Fourier transforms of one another. Unfortunately, and this is why there is X-ray crystallography as a science, if we can say, is that the phase is lost during the experimental measurement, and it's the most important component of the structure factor. So solving a crystal structure, once you have your diffracted intensities, you can only get from them your structure factor amplitudes, and you need to find the phases to generate an electron density map from which you can build your structure. And now we go a bit to a more practical approach. And in 1954, Max Perutz and some collaborators published the first method of experimentally determining phases from protein crystals. And it was a method which became known as the heavy atom replacement. And if you react a ligand containing a heavy atom, for example, a heavy atom means it's got a high atomic number. For example, containing mercury to the molecules of the native crystal, there are small changes that are introduced by this ligand to the diffraction pattern, and then you can use those changes to solve the phase problem. Why does that work? Because if you now think in terms of vectorial sum, you have the vector structure factor of the derivative is the sum of the vector structure factors of the native protein and of the heavy atom contribution. And they derived a way of going back from knowing the positions of the heavy atoms to the phases of the protein, and using something called the Harker construction. where you, once you know the contribution of the heavy atom to your structure factor, you can calculate back the phase of the protein structure factor. But as you can see here in this diagram, you have two possible values. So if you only use one derivative, you always have a phase ambiguity. It can be lifted under certain conditions. I will not go into detail. But if you have a second derivative which binds in a different place, then you can resolve this ambiguity in favor of one of the two possibilities. So, and now let's go to start talking about structures after this long introduction. Here you can see the first two protein structures that were solved by X-ray crystallography using the isomorphous replacement method. And they were solved more or less at the same time by both Max Perutz and John Kendrew. And here you can see them holding their high-tech models at the time of their protein structures. And they were awarded the Chemistry Nobel Prize in 1962 for this achievement, which was really a major landmark in protein structure biology. So here you can see in the 1960s, how people used to build protein structural models. So on the right, you have a scientist called William Lipscomb, which was using a contraption called Richard box. In the Richard box, basically, you would plot the sections of electron density of your protein in large plastic sheets. You would have them backlighted. And then in the front, you would build your model using tiny Lego pieces shaped like amino acids in what you thought was the right position. So this took a long time, and it was prone to errors. When I joined the crystallography department at University of Pittsburgh in 1983, people were still using this to build models of the crystal structures. And the joke around was that there was once one scientist that already solved the same structure 3 times. So as you can see, it was not very easy to use. So in terms of advances, in crystallization methods, we now have a vapor diffusion as a routine tool that is used for obtaining protein crystals. We have commercial crystallization screens that allow us to sample the different parameters that induce crystallization in many, many different ways. We have robots that can use small volumes and very reproducibly can generate the crystallization screens. And we also have automated visualization systems to replace human observation with its inherent error. And we have more advanced X-ray sources. So at ITQB, we have a microsource X-ray tube, which runs at a 50-watt power. So it's the light bulb, basically replacing the previous rotating anode generator that required 2.7 kilowatts of power at maximum intensity. And still, it was not as bright as this X-ray source. And then, of course, we have synchrotron X-ray sources. This is a diagram of the SRF. And you have much, much more the X-ray beams are much, much stronger. And indeed, you have the advantage that you can, in several beam lines, you can actually select the energy of the radiation that you want to use. In terms of detectors, so this is one of the very early detectors for protein structural biology. You can perhaps, so there's a rotation camera, and then you have 8 films in a rotating cassette. So basically, the crystal would be exposed for a certain period of time, and then the cassette will turn, expose a different film, and after you run out of films, you have to take out everything, go into the dark room, develop and replace the films with a new batch to continue your data collection. So I did that a couple of times at the SSRL when I was a postdoc at University of Berkeley, California, Berkeley, and I can tell you it's not fun. And then, of course, you have to digitize the film and go through all the processing, which is, again, it's not fun. And now In the late 1980s, we have advances. One was the imaging plate detector, where you can collect, you can read out the image automatically, but this took a long time to read the image compared to the time it took to expose, so it was not perfect. More recently, we have CCD detectors, pixel detectors, which are very, very fast. So nowadays, data collection at the synchrotron, in terms of the time exposure, is like 10 or 15 seconds altogether. So it takes longer to mount and unmount the crystal than to actually collect the data. And then, of course, we have crystal cryocooling, which helps reduce the radiation damage that's induced in the crystals by the X-ray beam. When you want to build and visualize, so remember that you saw the Richard box. In the late 1980s, the first graphics workstations appeared. They were horribly expensive. So this is an Evans and Sutherland. It's one of the first ones that came out. Then later, the silicon graphics machines came out, and they were slightly cheaper, but not this particular example. And finally, thanks to advances in hardware, in computing hardware, you can now buy a graphics workstation for less than 2,000 euros to work in your office. So as you can see, this was also tremendous, not only in terms of visualization, but also in terms of computing power needed to analyze and to continue your structure analysis. And then, of course, we had graphics display programs that go with these machines. Again, 3D stereo, I'm not mentioning here in detail, but it's also very important, at least for me, although some people don't like it. You can actually visualize the structure and the electron density map in 3 dimensions when you're working with it. So what happened since the time of Perutz? So basically what all of these advances permitted was to work with increasingly more complex molecules. So hemoglobin was very simple compared to this photosynthetic reaction center. And this is just a few examples. Then we have the GROEL-GROES chaperoning from E. coli in 1997, and the ribosome in the early 2000s, which so Huber and Jonat got the Nobel Prize for their achievements. Unfortunately, Paul Sigler was not so fortunate, but his work is also worth mentioning here. So, but we have recently, we got into another realm, which is the cryo-electron microscopy. And this is going beyond X-ray crystallography, because X-ray crystallography is limited for some cases because you need crystals. But for many interesting proteins and protein complexes, you cannot get crystals, no matter how hard you try. But if you're lucky, and you can use electron microscopy to do it. And we were also fortunate that we had a tremendous advance in cryo-electron microscopy, which went from the science of globology, as you can see in the far left image, to where you actually get electron density maps that are very close to the ones you can get from X-ray crystallography. And here the advantage, of course, is that you don't need crystals. You only need a pure sample, and not much even. So this is just a description of the technology. And here you can also see in a different diagram from the early microscopes to the most recent Titan Krios machine, which is horribly expensive, but it does give you these beautiful data that you can get. So how did this come about? And I'm almost finished with my presentation. First, Jacques Dubochet developed a technique called water vitrification. So we already had cryocooling for crystals, so he extended that to protein samples, and he used liquid ethane as a more gentle, fast cooling medium than pure liquid nitrogen. So the idea is to immobilize the molecules in a thin layer of amorphous ice. And you do that by applying your sample to a grid. You blot the excess, so you remove the excess solution so that you have a thin film only of your sample. And then you dip it very quickly into liquid ethane using this kind of device called a Vitrobot. And here you can see in the grid holder for the EM grids. So the advantages are that the molecules keep their natural shape, and the microscopes work in a vacuum. So when you put your sample in a microscope, it is exposed to high vacuum. So if you do that with a cryo-cooled sample, of course, you will have a little bit of sublimation from your ice sample, but it will be protected against sample dehydration. And also, to some extent, it reduces the damage by the electron beam. And then there was another major improvement, which was the development of directed electron detectors. And then you record movies instead of individual images. And then you can isolate the frames within each movie. You can do lots of tricks in the computer to make different types of corrections. And you isolate the different images of your particle, and then you group them together according to orientation. And in doing so, you can even separate different forms of your structure that are present in the sample. And there's some very nice examples of that in the literature. And this was the work of Joachim Frank and Richard Henderson. And as a result, these two gentlemen received the Nobel Prize of Chemistry in 2017. And the award was given for developing cryo-electron microscopy for the high resolution structure determination of biomolecules in solution. So for crystallography, the problem is getting crystals of larger and larger molecules and complexes. For electron microscopy, it's the opposite. It's getting structures of smaller and smaller molecules, because we have a limited amount of exposure that we can give to our samples. Otherwise, there's radiation damage. There is still a limitation in how fine we can view the pictures. And we need to see the individual molecules in the images, otherwise we cannot do any kind of reconstruction. So although the smallest structure that has been determined to date is IDH with 93 kilodalton to 3.8 angstrom. But it is expectable that more developments in recent years, in the years to come, will allow us to break down that barrier even further. And so that in the end, you can collect microscopy data of molecules as small as the ones we can study by NMR, for example, or X-ray crystallography. Another advance was to use electron microscopes to carry out electron tomography. And the advantage of tomography is that you can image cells and you can take a three-dimensional picture of what's inside a cell, and then you can image and you can reveal the organization of various components. And here, of course, this is a processed image. It's not how it comes out of the microscope, but then Julia Mohammed from Heidelberg, EMBL Heidelberg, is one of the leading scientists in this field, and this is some of her work. And finally, there's another recent development, which is micro-ED, microcrystal electron diffraction, where we can use an electron beam not to take an image, but to actually record the diffraction pattern of very, very small crystals, which cannot be measured. They are too small for standard X-ray crystallography and too large for X-ray lasers, XFELs, which are the strongest X-ray sources that are known. So this was developed in the United States in the lab of Tamir Gonen in UCLA. And this is just a slide to show you what you can do out of these very small crystals. And I will thank you for your attention.
Thank you very much. Thank you very much. Sorry?
Too much time?
No, that's fine. It's wonderful.
Okay.
Thank you very much for this fascinating journey through the developments. We'll move on to the next speaker now. Oh, yes. Vai ficar aqui? Não, se quiser.
Não sei. Eu estou, não sei.
Okay, so the next speaker is João Moraes Cabral. He comes from the University of Porto. He did several postdoctoral studies at the Dana-Farber Cancer Institute at Harvard University, at University of Leicester in UK, and later at Rockefeller University in New York. Joined the Faculty of Molecular Biophysics and Biochemistry Department at Yale University, and established his independent research group there. In 2008, he returned to Portugal as a principal investigator at IPMC and now at I3S, two prestigious institutions of the University of Porto. I was fascinated by the quality of his work when many years ago I visited his institution and he was kind enough to show me his structural work on potassium transporters. Please, well, tell us about the structure biology of potassium transporters.
So yeah. I see the email from Elena. I was so excited to be invited to speak at this institution that actually I didn't read the email completely and didn't realize what Elena was asking for me to do. So, but she was kind enough and patient enough that we could actually sort of bring it down to this very large theme, which is structural biology, potassium transport. And I've decided that the way to do it, to start, is to actually, sorry, I want to show you this. So this is just a sample of the many, many different structures that are deposited in the protein data bank that are involved on transport of potassium. And it includes potassium channels and transports. And so obviously these are membrane proteins. They reside, and this is a beautiful EM image of a muscle cell, a mammalian cell. They reside in these structures here, the membranes. And in this particular image, you can actually see many structures forming membranes. These are membrane proteins. They will reside on the membrane. And because of the physical chemical properties of the membrane, they act as barriers. So these membrane proteins facilitate the movement of potassium across the membrane, which is otherwise impermeable to potassium. But why is there such a large number of molecules that are dedicated to the transport of potassium? We're talking about potassium ions. And this is because all cells on Earth accumulate potassium. In fact, I would dare to call it a rule of life that all cells accumulate potassium and extrude sodium. Okay? And in fact, this aspect of accumulation of potassium in evolutionary terms is very, very interesting, because how has this emerged? If all cells have accumulated potassium, it suggests that very early, the evolution of the first cell was in an environment that contained large amounts of potassium. And there's actually not that many environments where that happens. So in terms of evolutionary questions, this is a very interesting aspect. But why is potassium important for the cell? In fact, high intracellular potassium is a base condition for cellular activity. The structure or the conformation of the genetic material is affected by high intracellular potassium. The structure of the function of the ribosome, the machinery that translates the genetic information into proteins, depends very tightly on having a lot of ions. So potassium is an intrinsic is a base condition for cellular activity. Additionally, potassium has a very important function in mammalian cells. And this is shown here. It actually establishes this difference, the fact that the cell is electrically charged is actually more negative inside than outside. And this is due to the fact that there are concentration gradients across the membrane in a mammalian cell. There is more potassium inside than outside, and there's more sodium outside than inside. This difference in this concentration gradient leads to the charging of the cell, so that is more electrically negative inside and out. So these gradients and this difference in electropotential is the basis of electrical activity in our cells. And so we have here a beautiful drawing, representation of a neuron by Ramon and Cajal. Here it is. These are the dendrites. This is actually the cell body or the soma, and there's this long axon that extends. We all know that the electrical impulses will run along the axon, and these electrical impulses are at the basis of the fact that I'm speaking, the fact that we see a lot of high-level activities are dependent on this nervous impulses. And here I have a modern representation. Actually, let me see if it works. In this case, it's actually a video from, it's a video from a colleague of mine, Paolo Ghiari in Trezes. What we have is a series of neurons. There's a soma, okay? A series of neurons and axons that extend. These sort of black dots are electrodes with which he excites and stimulates these neurons. And you can see that these neurons are flickering. There's light. There's a sensor that there's a small molecule that senses changes in membrane potential, that electrical potential that I mentioned. And when it goes to positive, due to the action, to the release of potassium and entry of sodium, these will, there's a nerve impulse and a depolarization that is shown here. Now, the important thing is that this process is very, very rapid. Notice at this time scale, okay? And so we actually do not see the propagation of the light across the axon because it's on the millisecond time scale, and the video is not does not have the required time resolution. But I want you to keep in mind this sort of, we're talking about seconds here, okay? And we'll come back to it. So what happens in this process is that we have channels. So some of those structures that I showed you are the membrane proteins that are in the membrane. And so we need the action of potassium channels and of sodium channels that will, when they are open or active, stimulated beyond a certain threshold, they will let sodium in and potassium out. And here we actually see a representation of the action potential. The light that we saw in the movie was when it attained this sort of level. This is membrane, this is potential, this is time. And when the light, when the potential at a certain level, we saw light. And so you can see that after a certain stimulus, there's a series of failed initiations and were these little flickers that we could see. But in some events, it goes beyond a threshold, the sodium channel is open, sodium rushes in, depolarizes the cell, so it becomes more positively charged, and then it activates the potassium channel. So potassium will go out, repolarizing the cell and bringing it back into a negatively charged state. So this is called the resting state. So the potassium channels and potassium have a role in maintaining the resting state, and they have a role in the definition of the action potential. Now, when we have this effect, when the channel is open, it is obvious that you will lose this concentration gradient. So there's a dissipation of these gradients. And therefore, you absolutely need another membrane protein that the sole function of which is to transport potassium in and sodium out. And this is called the sodium-potassium pump. It is actually amazing to understand that the brain spends as much as 40 to 7% of its energy powering this machine. A large amount of the energy spent by the brain is to maintain those gradients across the membrane. So the sole function of this is actually to build these gradients, more sodium outside than potassium in potassium. Okay. So in a way, we have all these collections, these very different shapes and very different channels, because we need to tune the properties of the channels so that it will affect their properties. One other thing that is surprising, and it's buried in the middle of all of these structures, is that the ones that are highlighted with the spheres are actually bacterial proteins. And these bacterial proteins were actually the first, among the first and most, among the first to be solved. And we learned a lot about eukaryotic mammalian potassium channels from the bacterial system. But what was always intriguing was, why is it that bacteria have these proteins if they're just bacteria? Why do they need at all proteins that apparently are involved in very high functional, highly elevated functions in mammalian cells, which nerve in? And so this question has lingered around, okay, until very recently. And here I have another video from the Suella that shows something quite extraordinary, okay? And I'm going to show you the video. So this video is of a bacterial species called Bacillus subtilis. What we actually have is a, oops, sorry. What it actually is, again, I lost it. What this actually here is a biofilm. And the biofilm, like the neurons that I showed you before, has been labeled with a small molecule that senses changes in electrical potential in the cell. And so if we start the video, oops, we have this. And what we see here is an expansion of the biofilm. So the biofilm is a collection of bacteria. It's a very particular collection because cells are differentiated. They are genetically identical, but they have differentiated. They form what we could describe as a proto-organism. What we see now in the film is, I'll show it again, is that the biofilm expands, the cells are divided. And at the same time, we see changes in the color across the biofilm. So I'm going to show you. Okay, so you can see what happens in the process is actually there is lighter outside, for example, this space, and I'm going to start the film again. And what you'll see is the propagation of the light across. So there is a at this moment, these cells in the periphery are more depolarized, and this effect will be transferred across to the center. Okay, so just a second. I'll continue. So what we're actually seeing here is electrical activity. This is reminiscent to what we saw when we were looking at the neurons. So bacteria are able to have electrical activity. The hypothesis at the moment is that this effect is a form of communication from the cells in the center of the biofilm. This biofilm is has a very dense matrix. This is a form of communication from the cells in the center that lack nutrients to the cells on the periphery, so that they will let nutrients pass through the biofilm and feed the cells in the center. Now, this is just an hypothesis, but it is nevertheless the first demonstration that, a very fantastic, amazing demonstration that bacteria are able to perform electrical activity and in a coordinated fashion. Okay, and these processes involve potassium fluxes. So this explains why do bacteria have all this collection of bacterial potassium transporters, which we have used very efficiently to solve structures and learn about potassium channels and transporters, but have not yet understood what they do in the organism. Now, we became interested on this process, and in particular, what we're looking at is at understanding how the machinery in a bacterium coordinates its activity. And what the system that we use is the Bacillus subtilis. It's a beautiful gram-positive system. All the components of the potassium machinery are known. There's channels. These membrane proteins here include channels, and I'll be talking about these two here. They're called, they function as potassium importers. Here we have two sets of potassium proton antiporters that work as potassium exporters, and there's a single potassium channel which is involved somehow in potassium release, and it's known actually is involved on that phenomena that I just showed you in a bio. There is also worthwhile mentioning that in bacteria, potassium does not really have the exact same functions as in mammalian cells. In particular, it does not set the membrane potential. It modulates the membrane potential, just modulates, just sets its point. It has a role in regulation of intracellular pH. It has a role in regulation of cell turgor. These Bacillus subtilis, they have 14 atmospheres of internal pressure, and potassium is involved in it. It is involved also in environmental adaptation, and now we know somehow they're involved in cell-to-cell communication. Now, what we've known about the Bacillus subtilis machinery is that there is has been proposed that there is a secondary messenger, this molecule here, which is a bacterial messenger that does not exist, it does not function in mammalian cells, that acts as a master regulator. And it has been shown, it was proposed, that it would activate the exporters and inactivate the importers in a very simple model, just like a switching on and switching off of the machine to control the levels of potassium inside. Now, we've been looking at this in more detail, and in particular, we focus on this antiporter down here, one of the potassium. What we've done is this protein, membrane protein, is actually composed by two proteins, a membrane protein I showed you here in blue, and a regulatory subunit. We have used structural biology to determine the structure of the regulatory subunit and to demonstrate that and here's the structure, that in fact it does bind cyclic AMP. This was just a proposal before. We now demonstrated it does bind the secondary messenger. Using a in vitro functional assay where we basically reconstitute, express, extract, and purify the membrane protein and we can actually measure its activity. And we could see what the effect of cyclic AMP is on the function of this export. And here it is. So this is a fluorescence assay. This is time. This is fluorescence. There's a first initial phase of the decay just to set up the assay. And then when we add potassium at this point, 200 seconds, we see a dequenching in fluorescence. Okay. Now these curves here correspond to the curves for without nucleotide or with a series of nucleotides that I showed you here. Okay. But when we add cyclic AMP, we can clearly see a faster dequenching, demonstrating cyclic AMP is the activator. This molecule is controlling the activity of the exporter, supporting what had been proposed. This is the demonstration, quantitative demonstration, that that molecule, cyclic AMP, activates the exporter. Now we went beyond that, and we actually determined many different curves, okay, at different pHs, with increasing concentrations of cyclic AMP. So the darker the color, okay, in any of these graphs, the more cyclic AMP we have in the solution. And what you can clearly see is that on pH 8.5, the curves are have a larger amplitude, and they change in more rapidly. And so we can convert these curves into rate constants. So we measure the velocity of change, and we can plot them according to different pH. And so here's cyclic AMP concentration, and this is the velocity of the change. Okay, and we have one curve for each pH. So we can clearly see several things. First, the basal function, the basal activity without cyclic AMP increases with pH. But more interestingly is that the midpoint, which is given here by this K1.5, shifts to lower concentration of cyclic AMP as you increase the pH. So the system is, the activation by cyclic AMP is highly dependent on pH, okay? The system is much more sensitive to cyclic AMP at high pH, we have a K1.5 of less than a micromolar, than at pH 7. At pH 7.5, which is the normal, close to the normal pH, cellular pH of a bacterium, of a Bacillus subtilis, you actually would have very little or no activation by cyclic AMP. Regulation by cyclic AMP would be almost inoperable, okay? And so we went a step further, and we actually went back into the bacillus. And these are just graphs showing time and growth of the cell in different conditions. You can see, and we made a mutant so that we actually removed the antiporter, but we brought it in back through a plasmid. This is just with a membrane protein, the two components, or a mutant that I'll So a pH 7, with or without potassium, there's no effect. They all grow equally. The curves are identical. A pH 9, though, just a second, a pH 9, though, when we have no potassium in the medium, okay, the cells do not grow. But if I bring potassium into the media, some of them grow, others don't. In particular, when I have the membrane protein being expressed, it complements the phenotype. The cells are able to grow, okay? When there is the same thing happens with the full complex, with the empty plasma, it doesn't grow. And more importantly, is what happens here. We had constructed a mutant, which we had learned, understood from the structure, which is this one. It is this arginine that interacts tightly with the cycloxanthine. We demonstrated that if we, from the structure, we learned that if we mutate this, we would lose cycloxanthine binding, and we, in fact, we demonstrated it. And what happens in our assay? There is no growth. So this actually is a novel idea that we're proposing, is that suggests that cycloxanthine not only has a role in controlling the machinery for potassium, it actually has somehow a role for controlling the intracellular pH. This is something that is new. All right, so what about the importers? So the proposal says that these importers will be switched off by cycloxanthine. We know that these importers are the KTRA-B and the KTRA-CD. These are very conserved membrane proteins in bacteria. We actually know a lot. We saw the structure of KTRA-B quite a few years ago. It has two components, the membrane protein KTRA-B, the regulatory subunit KTRA, which forms these, viewed from the bottom, forms these beautiful rings, right? Each one of these 8 subunits is an homo-octamer, and each subunit is shown in red or blue. We know that it binds ATP or ADP, and using a similar in vitro system, we actually can demonstrate, using fluorescence flux assays, that ADP gives rise to a little bit of activity. So ATP activates a little bit of the system. ATP activates a lot. There's a clear dequenching very, very fast. So ATP acts as an activator. It's essential for the activation. Now, there's one thing that is interesting is in KTRA-B. I showed that there were 2 flavors of the KTRA channels. There was the AB and the CD. Since AB form are arranged, genes are arranged in an operon, it has been proposed that it would form the AB pair. And by default, the C and the D should form a CD complex. Actually, through biochemistry and other means, we've demonstrated that we can also get the regulatory subunits A and D are actually very promiscuous. So we can actually form CB and AD. Moreover, since AB, the regulatory subunits are so closely related, they're 50% identical, we can also form heterooctamers, and we have the possibility of forming these KTRA, CB, and CD. So this actually, through our work, we actually demonstrated that the system is much, much, much more complex than previously. And we have evidence now from the cells that, in fact, some of these potential complexes exist in the cell. And this is shown here. So this is in Bacillus subtilis, we basically grow in Bacillus subtilis overnight in different concentrations of potassium, and this is growth. So we can see that Bacillus subtilis wild type requires 0.3 millimolar of potassium to grow well. Okay? At 0.3 millimolar, they grow fine. If we now make the deletion mutants and remove B, which is one of the membrane proteins, we see this. Without B, which is a major potassium importer, we need at least 2 millimolar. If we bring B back into the system, we completely recover phenol. On contrast, if we remove D, the other membrane protein, we see nothing in this house. Okay? So B is essential. B is essential for Bacillus subtilis to grow at low potassium concentrations. It acts as a lead. What about the regulatory subunit? Well, if we remove A, we don't see anything. And that's surprising because I have told you the proposal was that A and B will form a complex. And A is essential for the activity of B. We know that. So deleting A should have resulted in a phenotype that will be identical to this one. But that's not true. However, when we delete C, which is the other regulatory subunit, we see this. And notice actually the change in the scale. Okay, Without C, we require 30 millimolar of potassium. So C is controlling the C, deletion of C is equivalent to deletion of both A and B and D, so both membrane proteins. C is obviously controlling all of them at the same time. So this demonstrates that C not only associates with D, as it had been proposed, but it associates with B. So it's the first demonstration that some of these complexes exist in this. Okay. What about cyclid-IMP? I sort of left it to this. Now I'll be finishing very soon. What does it do in this system? It was proposed that it should inactivate this. And here's the flux assay, very similar as before. And what we have here is the activity of CB and ABAP. Okay, so here the thing. There's the activity of B, slightly less. When I bring cyclid-IMP into the system, here it is. Cyclid-I switches on the CB, but it does not switch off the complex that is reconstituted with it. So cyclid-I only switches off the complex that has C and not A. Once again, supporting the idea that C is the major regulatory system. This is just a quantification. What about those heterooctamers? Are they also inhibited by cyclid-IMP? And the answer is yes. Whenever there is C, the system gets switched off. Okay? So what I've shown you, and this is the last slide, what I've shown you is that the system is really, the bacterial system in terms of potassium transporters is actually very complex. The regulatory network is also fairly complex. It has, cyclid-IMP is this molecule here shown here. There's many potential. In low potassium, we know that we have low cyclid-IMP concentration, so all of these importers are active, and the exporters are probably inactive. We also know that cyclid-IMP controls the activity of transcription of some of these, and in high potassium, okay, I'll just skip here. In high potassium, we have an increase. Oops. In high potassium, I don't know what happened. Yeah. In high potassium, the cyclid-IMP will increase. It will bind to some of these, the ones that are controlled by C, switch them off, and it will at the same time activate the exporter. But all of this, this is the simple image. The truth is that since the activity of this one depends so much on pH, this image is not sufficiently clear, and much more work has to be done to understand how the potassium transporters in bacteria work to maintain the physiology of bacteria. So I need to thank the people that did the work, Celso, Rita Rocha, Tatiana, and João. I need to thank, of course, two people at ITQB, Monica Serrano, Adriano Enrique, that were essential for me to learn how to do genetics on Bacillus subtilis. And I need to thank the funding bodies, in particular FLAD, more recently La Caixa, also FCT. And now I have to say one thing, and Elena will forgive me for that. Very recently, we've learned that, and this is in life sciences, that 4 Portuguese scientists were awarded the ERC Senior Advance. And this is, of course, a breakthrough in a way for Portuguese science. It took us 30 years from the beginning of the development of the growth in science in Portugal to have scientists that are technically and particularly conceptually at the forefront of European science. My alert, though, is that FCT, and this is important because we're in this house and we need to say, FCT has clearly changed its focus. And though we have to thank FCT for the continuous support to Portuguese science in the past 30 years, more recently, that support in terms of doing and making science has decreased drastically. It took 30 years to get to this stage. It will be very fast to deteriorate very rapidly if FCT does not refocus funding in the proper fashion. Okay, thank you.
Thank you. Very well. Thank you very much for this wonderful talk and for coupling structure and physiology. She is very exciting. And now we'll move to someone who is now not, who is in German. Oh, well, so we have Anna Messias via Zoom. And I'm sorry because I think I assigned you a very difficult job to talk about NMR in this kind of environment. So exciting. But that's life, and I'm sure that you'll do a good job. Anna Messias obtained her PhD in 2000 under the supervision of Professor António Xavier, and I was very happy to be invited by him to be her co-supervisor. She was part of the team who solved the first complete 3D structure of protein using NMR in Portugal. She completed postdoctoral work at the MBL in Heidelberg, and then she was a research fellow at Sanofi Aventis Corporation in Frankfurt, using NMR for drug discovery. In 2007, she became a group leader at Max Planck Institute for Molecular Physiology in Dortmund, established a group on structure-based drug discovery. Since 2010, she is the leader of a team in the Helmholtz-Zentrum Mund in Munich, focusing on metabolic and neurodegenerative diseases. So she has been very active all these years, and I'm very pleased to hear you about NMR spectroscopy of proteins.
Thank you. So I hope that this will work out well. Can you hear me well? I hope so. I can't see. Let me see. Yes. So I would like to thank Professor Lena Santos for giving me the opportunity to share a bit of what I have done since my PhD. Actually, I started using NMR before my PhD. And today I will talk about NMR spectroscopy of proteins, its application on structure, dynamics, and interactions. And I have to start by maybe explaining a bit what is nuclear magnetic resonance spectroscopy. One important point is that it's a very informative technique, and also that it's a non-destructive analytical tool. This is basically the only NMR that I will say. It uses the inherent magnetic properties of specific atomic nuclei under a strong magnetic field, which absorb or release energy at radio frequencies, and we use then this energy to reveal the molecular behavior. And we do that for different nuclei, which are what we call NMR visible nuclei, as for example, protons, carbon-13, 15N, and 31P. An important point, because sometimes people get a bit confused. So all these visible isotopes are non-radioactive, which makes it a very simple and non-hazardous technique to use. So basically what we do, we put our samples in tubes, and here I have some of these tubes so that you can see. They are small tubes, with the width of, for example, 5 millimeter, 3 millimeter, or recently of 1.7 millimeter. And then we record our spectra. And then typically in NMR, what we have different dimensions. And the more dimensions we have, then that means that we are acquiring more complicated information. And here's the simplest spectrum that we can record, which is a 1D.
And I'm sorry to interrupt you. We are having some problems with the sound. Can you please turn your sound a little bit up? Because it's on your side, not on ours.
Ah, I see. Let me try. And now
I think it's better.
it's better?
It's better.
Okay. I think now it's at the limit. I can't put it higher. So I hope it will be fine. I can move the microphone a bit closer. So I was explaining you that the simplest spectrum you can record is a proton 1D. And here you can see how such a spectrum looks like. So basically each one of these peaks here, it represents a chemical group. And this is a 1D of a protein. And from the dispersion, we can already judge how well the protein looks like in terms of folding. And also what is very important for biochemists and chemists is, of course, that depending on the region where the signals appear, it will tell you about the type of groups that you are looking at, and we'll use that information intensively when we are studying molecules. And then from 1Ds, we can move to 2Ds, 3Ds, 4Ds, depending on what we want to look at. And what we record frequently, and I will show this very often during this presentation, is what we call a proton-nitrogen correlation spectrum, where you, in this case, for a protein, we'll look at the resonances of backbone amides. And this is like a fingerprint region for proteins, which makes them very, very valuable. And as I said before, in NMR, we use strong magnetic fields, and that's most of the times how NMR spectroscopists are known, because we use very big machines. And some of you might know also magnetic resonance from this kind of machine, which is not the kind of machine I'm going to talk about today. This is a very big machine used for imaging of medical conditions in a clinic in Munich. But we actually use other big machines, but look different. And here you can have a look at our NMR spectrometer hall at the Bavarian NMR Center, so here in Munich, where we have, I think at the moment, like 14 of these machines ranging from very different fields. And when we're talking fields, about fields in NMR, you can talk about Tesla, but we like, when we, in spectroscopy, we like more to talk about the frequency of the protons. And that's why we talk about a 250 megahertz spectrometer, 1.2 gigahertz spectrometer. And there have been really tremendous changes and developments in the NMR spectroscopy field. And for you to have an idea, when I did my PhD, that's where I spent quite a lot of time, is using such a spectrometer like this one, 500 megahertz, which at ITQB was top of the top. And I was really very, very happy to have the opportunity to use such a machine. Nowadays, here in Munich, we just acquired the 1.2 gigahertz. And this has changed dramatically the kind of studies we can perform, particularly in terms of sizes of proteins. And in the next slides, I will talk a bit of what I have done in the field of NMR, and also show you how versatile this technique is and how much you actually can do with it. So as I told you, I started my PhD at the ITQB with Professor Lena Sanch and Professor Antonio Xavier. And at the time, this was like the years where NMR was starting to be considered as a major technique for solving 3D structures, and I really, really wanted to learn this. So I was extremely happy when I had that opportunity to do it at the I2QB. And here I'm actually showing my first structure that I solved by NMR. So these are this bundle in white. And you can see how beautiful. I find these structures extremely beautiful, also because you can see for the tetraheme cytochrome C3, it has the hemes, and it's basically just hemes with a bit of protein, like I like to describe. And it was a big privilege. I really learned a lot during this period. And I also started to learn how to interpret these structures, how you can, from a 3D structure, you can relate function of the protein, like in this case, how do you relate like the transfer of protons and electrons with these conformational changes. But I wanted to learn so much more, and I had the opportunity to move to Germany, and I moved to the MBL in Heidelberg, and I joined the group still very 100% focused on NMR spectroscopy, but applied to other proteins. So in this case, the group was very, very focused on protein RNA interactions, and in particular on splicing. And I worked with a protein called splicing factor 1, where I used NMR again to solve the 3D structure of this protein with RNA, and we could interpret actually very, very well how the protein could recognize specifically the sequence, in particular the branch point sequence. But more than that, because we had the structure of the free protein, we had dynamic studies, we could also understand what happens to the protein between the free and the bound state. And it was extremely interesting because, for example, this helix here, which is barely seen in the free state, and is very flexible also in the free state with nanosecond to microsecond motions, actually folds upon binding to the RNA, and then it also rigidifies. And that's something that we can do by NMR. So we can change how the motions of the proteins change upon different conditions by, for example, binding to some small molecules or other proteins. And after my postdoctoral work at the MBL, I continued being very, very interested on NMR, but I wanted to move more in the direction of drug discovery, and in particularly of focusing more on medically relevant proteins and how we could use NMR spectroscopy for drug discovery. And in the next slides, I will give you some examples of studies I have done in that direction. And here I'm showing some images about studies that we have done in the NF-κB pathway, which is a very important pathway in inflammation, and how we really tackled the analysis of some interactions and analyzed small compound interactions. And one of the strengths of NMR is that, for example, we can make one protein visible by NMR by using visible isotopes that are visible by NMR, then add other proteins which interact with it, and just study the system just looking at one component or the other component. And in this case, we were looking at the nemodiubiquitin system. So we looked first at the diubiquitin, so we labeled it with nitrogen 15. We recorded the 2D, and basically here you see this fingerprint region where each dot is a backbone amide. Because we have the protein aside, we also know for each dot to which amino acid it corresponds. And then if we had the target, in this case, NEMO, which you follow in blue, you can see what happens. So if the spectrum stays the same, nothing has happened, they do not interact. But if you see things happen, that means that they are interacting, and because we know very well which dot to what it corresponds, we can map it on the structure, and that's what we have done here. So basically, we mapped the interaction of NEMO with diubiquitin following the diubiquitin site, and we did the same following from the NEMO site, which is this long coil curl. And when we added diubiquitin on the other side, then surprisingly we saw changes all over the protein, and this told us that in the case when we titrate diubiquitin to NEMO, that there are more than a binding event, there is also a structural rearrangement of NEMO, and that this prevents the binding of a second molecule on this site. And that's what you see here. So there was a molecular model where there were two diubiquitins binding to two NEMO molecules, but actually in solution, what we know is that only one is available to bind because of this structural change. And we moved further because our collaborators wanted actually to find inhibitors targeting this protein-protein interaction. So they found in screen some hits, and we tested them by NMR. And we can do that also very easily in NMR if you build the complex. So you are looking again at the complex, and then you add the molecule, the small molecule. And if you see changes again, then that means that the compound is disturbing this interaction between the two proteins. And we worked further also to characterize the binding, and we could determine that the small molecule was interacting with NEMO and not to the diubiquitin. And one other study that we did, and just to put you in perspective, where I'm working now at the Helmholtz-Zentrum München, diabetes and obesity is one of the major things, and we are also very focused on small molecules from natural sources, and celestrol has come recently as one of the most potent molecules that could be used for targeting obesity and type 2 diabetes. So we wanted to identify possible targets that could be relevant and then in drug discovery for this molecule. And using this same kind of procedure, what we did is we added celestrol to protein tyrosine phosphatases and could identify two tyrosine phosphatases specifically located in one part of the brain called the hypothalamus, where the compound has a major effect. And then we used also NMR, specifically using competition experiments, just in 1D experiments, where we could show that celestrol is a non-competitive binder, which does not bind to the active site of the enzyme. And now in my 10 minutes that I have, I would like to talk about a project that lies deeply in my heart because it is a project about a disease called MPAN, which is a very severe neurodegenerative disease affecting mainly children, and for which there is no cure. And this disease was actually identified at our center, and we have been working on it. And I find this one of the most challenging, but at the same time, one of the most interesting projects that I've been working with. And I will share now some of the ideas that we have and how important NMR actually was for the discoveries we have made on this protein. So mutations on this gene cause this MPAN disease. And when we look at the gene product, actually it codifies a very small protein, which is only like 150 amino acids, depending on the isoform. And we know also that is localized to the mitochondria. If you look at the sequence, actually you realize that the protein only appears in higher eukaryotes, and it's quite conserved, but it has really no homology or nothing so significant to other non-proteins. And though at the beginning we're very suspicious, this really told us from the beginning that we are facing probably a new protein fold, and that this protein has a new function. And typically when we get a new project in the lab, we clone it and we try to see by animal how it looks. And because it has this transmembrane region, we try to avoid it at first, and we just focus on the N-terminal and on the C-terminal domain. And actually we didn't know, so we knew that the protein was localized to the membrane, but it could be that one part was facing the cytosol, the other part more the intermembrane space, or even the matrix, or the other way around. So everything was open, and we started just doing simple NMR experiments. And actually I did these experiments myself. So I put these two constructs in the NMR tube, and I recorded some NMR spectra. And I was very sad because none of them looked really promising by NMR. The N-terminal part was basically unfolded. The C-terminal part looked folded but non-tractable to be used by NMR. And I was sad. And then I thought, why don't I just try to mix the two together? And actually, when I mixed the two together, suddenly we had what is called a very high resolution spectrum, and we could tell that these two parts should interact with each other and that they form the soluble domain. And because I knew that from the sequence that there was a cysteine, so it was clear also that a reducing agent could have an effect. And in fact, when I added reducing agent, the spectrum quality even improved even better. So we went further to label these peptides so that we could use bidimensional, three-dimensional spectra. And then, And so what these experiments, of course, told us is that the other two hypotheses were not clear, and that we have, of course, the N and the C-terminal part should be on the same side of the membrane. And here I'm showing this proton-nitrogen correlation spectrum, where you see that we have a very nicely folded protein domain that we could follow by NMR spectroscopy or any other technique. Recently, it was also, it came everywhere in the news that you could use AI, or specifically deep learning structure prediction methods to predict the structure of unknown proteins. And I had been trying this kind of methods for a long time, and nothing ever looked that promising until recently. And then when it was possible to submit the sequences there, I submitted the sequence of our protein. But I was very surprised that somehow especially the most famous, the AlphaFold, gave a very strange result. And then, of course, because we had the NMR data, we could verify if which one of these models actually made more sense and was more accurate than the others. And because we had the NMR data, so we had assigned the soluble domain, we could tell straight away, because of the presence of secondary structural elements, that the AlphaFold model is very inaccurate because it does not possess any strands, when we know that there are strands in our model, while the other models could be reliable. So we went further to combine this new tool and use it together with NMR, really to produce a soluble domain that could be used then, for example, by X-ray crystallography, so that we could solve the structure easily. And that's what we have done. So we basically just designed different chimeras of fusion proteins of the soluble domain, where we link these two parts here where the transmembrane region was with different linkers, and then followed by NMR, the changes. And the NMR in this case is very important because here we can really see if we are not destroying the fold of the protein and if we are maintaining the major features of the domain. And that's what happened. But unfortunately, there was still too much flexibility in this loop, and that's because we couldn't manage to crystallize the protein. So we went further, we reduced the loop more, and then deleted also the N-terminal part, and then suddenly we could obtain very good crystals that we used to solve the structure of the soluble domain. And here I'm showing the high resolution structure of this soluble domain, and I'm comparing it with the predicted structure, where you can acknowledge actually how similar it looks. And as I expected, we are in the presence of a new protein fold, which actually is extremely rare nowadays to find proteins with a new fold. But our task is just starting. And actually, though we have now the structure of the soluble domain, or what we want is the structure of the full-length proteins, so therefore included the transmembrane region. But when we include the transmembrane region, it starts getting even more complicated because the protein oligomerizes. And so we are following studies in detergent and in nanodiscs. And as usual, we always use NMR to make sure that we are in the presence of a native conformation. But what I have learned also over the years is that there are so many good techniques that we can combine with NMR spectroscopy, and we use everything that can help us answering our biological questions. And here you can see that we have been using X-ray crystallography on the full-length protein as well. We are using low-resolution techniques like SACs and SUNS, and also cryo-EM. So, and I'm almost finishing. Just to summarize, I hope you could see how powerful NMR spectroscopy is on the study of proteins, from the design of construct that you can use for in vitro and in vivo studies, for the studies of dynamics, so the studies of the motions of molecules, for the 3D structure determination, and now you can use that for structure function understanding, for interactions with small molecules, and these can be modulators, substrates, activity modulation, to interactions with other proteins, like identification of biological partners, and at last also drug discovery, where you can use NMR for drug screening and hit validation. Also, you can also use it for solving the structure of ligands and use that information to improve your ligand and hit candidate. So at the end, I would like to finish by acknowledging my path, all the mentors, all the students that continue to teach me really a lot. The funding, without it, we couldn't really do science. And my family, my husband and my children, who actually are always behind me and support me in any way. And I thank you for the opportunity to give this talk. Thank you.
Thank you, Anna. Thank you,
Thank you.
Anna. It was a beautiful work, and just a pity that you would never be able to do it in Portugal. But I wish you all the success.
Thank you.
And now we'll have Filippo. So, Filippo Mancia is a professor in the Department of Physiology and Cellular Biophysics at Columbia University, and I think this tells a lot about the quality of his work and his career. He's a membrane protein biochemist and structural biologist with experience in X-ray crystallography, single particle cryo-electron microscopy, and in production and characterization of membrane proteins. The main focus of his lab is on membrane protein lipid interactions. He has an outstanding curriculum and he owns an endless positive energy that I guess relates in some way to his Mediterranean ancestors. We look forward to your lecture.
So this year, I think the slides, we need to make them advance. Okay, so first of all, I'd like to, when our two hosts here, I'd like to thank Elena for the invitation and mostly for, for what's really been almost a decade of a fantastic collaboration. And I'll get to show a little bit of our joint work in a bit. I'd also like to thank Ana Maria Nuno for having organized this event. I'm honored to be here today, and it's a real pleasure for me to be able to present some science, some of the things we do. So let's start off taking a little step back in history. It was 1982, and it was the beginning of the summer in 82. I think, at the beginning of high school. I had no idea what a protein was, or barely, and I had limited interest in the whole subject. I think I was looking forward, like all Italians, in what was going to be a superb summer of 1982 in Spain for the World Cup. However, while the Italians were focused on that, the Germans were hard at work, and this really represents a bit the history of Europe as we know it. And in particular, the biochemist Hartmut Mickel in the lab of Robert Huber and together with Karl Deisenhofer was working on what was thought at the time to be an intractable problem, that of determining by X-ray crystallography the structure of a membrane protein. This was due to the fact that the membrane protein sits in a membrane. The biochemists have to use these heterogeneous detergents to extract it. So how could this be possible, that you could take this sample and have it form an ordered crystal? However, in the hilltop of Erice, in the northwest part of Sicily, in a conference in June of 1982, Hartmut Mickel stands up and he shows this. This is the diffraction pattern of the photoreaction center, the first membrane protein to have ever been crystallized. 2-3 years down the line, Hartmut, Karl, and Robert solved the structure of the photoreaction center shown here. This leads them the Nobel Prize a few years later. And this is considered the annus domini, the year zero of structural biology applied to membrane proteins. The field has, after about 10 years of silence, shown here on the right, witnesses a close to exponential explosion, first with the work of Rod McKinnon's group on potassium channels, and then Eric Gore with the transporters, and many others have contributed to a spectacular success of structural biology applied to those proteins that sit in the membrane. However, despite the advances, if we look at the statistics and we look at structural biology in general, they still represent about only 3 to 4% of all the structures that have been determined. So the intrinsic problem really goes back to the fact that the things sit on a membrane. And if you're going to determine their structure by X-ray crystallography, which is a technique up to about a decade ago, it was the most efficient technique to determine high resolution structure. X-ray crystallography requires abundance. Abundance in a membrane protein that sits in a two-dimensional layer, you just can't put enough of it. So you encounter problems at every step of the pipeline to produce the protein for structural studies. But the most important and detrimental part of all this is exemplified in this schematic here, where you have a protein that typically sits and lives its life in a bilayer, in head-to-head lipid interactions that form all biological membranes, and the membrane protein biochemists pretends or expects to be able to extract it from there and put it in a form in which the detergent, which forms the sphere around it, mimics to some extent that, but it doesn't mimic. So the loss at every stage of the process is absolutely tragic. And if you look at the graph on the upper left, on the upper right, you can see that of all the membrane proteins you try, only about a third of them you will be able to express them in the recombinant system to sufficient amount for structural studies. And then you lose another factor of 10 for an issue of stability in the transition from the bilayer to the spherical micelle. And then you've got a catastrophic other tenfold effect when you try crystallizing it. And the bottom line is that if you start off with 1,000 random membrane proteins, only about 0.2 to 0.3% will yield initial crystals, which are the requirement to obtain structures by X-ray crystallography. So what we and many others did was to focus on the first two steps and let nature take care of the problem, i.e. look at sequence variability within naturally occurring genomes, of which we have had an increasing number of sequence information, to find those variants or orthologs of a given protein of interest that could be sufficiently stable to increase the likelihood of obtaining structural information. The approach was called structural genomics, and I worked on this for the last 20 years together with Professor Wayne Hendrickson at Columbia in what was first the New York Consortium of Membrane Protein Structure, or NICOMS, and then the Center of Membrane Protein Production and Analysis, or COMPA. So we used a miniaturization of the problem. The idea was to spend as little money as possible and to screen as much as you could to identify those orthologs of a given candidate gene that would have high likelihood of yielding structural information. We roboticized as much as we could, and this is just shown a Sonnet 96 sonicator here on the left that is working in action, sonicating bacterial cells. And that just a result of this is shown in this gel, where for a given gene of a target membrane protein, you could see on the top gel and analyzed by Western blot analysis that only about 4 out of 25 orthologs. This is the gene that does the same things in 25 different bacterial species, or 24. Only 4 of them we could express in a recombinant system. And in the one in the bottom, we're down to 2, validating the use of structural genomics for membrane protein structural biology. And this has allowed us to tackle biological problems of substantial importance. And one of them that we tackled in the lab, and here I'm going to link it to the collaboration and to getting to know Elena, is the basic questions of how do glycerol phospholipids, how are they made, and what is the identity? How is it determined? How is their identity determined? The identity of a glycerol phospholipid is schematized there on the left, which has a glycerol backbone, 2 acyl chain, 2 hydrophobic chain that constitutes the bilayer, the glycerol phospholipid being the most abundant lipid in all biological in all biological membranes is due to that phosphate in that alcohol. The identity of the alcohol will determine if it's a phosphatidylcholine, it's a phosphatidylserine, or if it's a phosphatidylethanolamin, or inositol, or glycerol. This constitutes the basis for the synthesis of phosphatidylcholine and phosphatidylethanolamine, and by reflex of phosphatidylserine, of what's known in biochemistry world as the Kennedy pathway. The enzymes that do this job, that take essentially the phosphate and create a phosphodiester bond linking two alcohols, the one at the head, at the tail, at the head group of the glycerol phospholipid and the glycerol itself, are called CDP alcohol phosphotransphorases. And they work by having one of the two alcohols, shown here as R1, activated, linked to a nucleotide, which is, in this case, is CDP. So it's CDP alcohol plus what's called the acceptor substrate, which is the other alcohol. The phosphate comes from the CDP, which becomes from a diphosphate to a monophosphate, and you've put phosphate sensor stage, and you've made the last step in glycerol phosphosynthesis in glycerolipid phosphosynthesis, in glycerolipid biosynthesis. And shown there in the bottom part is the example used by eukaryotes and some bacteria of how phosphatidylinositol is made, where the activator, the CDP, is linked to the diacyl glycerol, that's the glycerol with the diacyl chains, plus myoinositol. A bit of magic happens when the enzyme comes along, leading to the release of the cytidine monophosphate and the phosphatidylinositol. So we became interested in this step, being interested in the lab, in processes of how the biological membranes are made. And we screened, take a step back. This family of enzyme is extremely varied because the CDP part, the one that provides, let's say, the energy for the catalysis to occur, can be on either side. It can be on the diacyl glycerol in some cases and on the terminal alcohol in the other. So there's various options and permutations which are shown there in the middle, and they all catalyze the same reaction. And all we knew at the beginning of this family of enzymes, this vast family of enzymes, is that there were there was a sequence, a conserved sequence of 8 amino acids, 5 on one side, shown on the left, and 3 on the other side, of 2 putative transmembrane helices. This constitute the signature sequences of the CDP alcohol phosphotransferases, and that's all we knew. So we and others went into this world trying to crystallize these things, and it was a complete disaster for a while because we screened and we just could not obtain any crystals. Until one day, in our structural genomics efforts, we were able to identify a subfamily of the CDP alcohol phosphotransferases that had an additional domain which carried an additional function. This additional domain that sits outside of the membrane and shown there on the lower part of the slide was critical to obtain crystals in a technique called lipid cubic phase, in which the protein is reconstituted in a lipid and it forms sheets of reconstituted membrane. So the fact that we could identify these subfamily of the CDP alcohol phosphotransferase with this additional domain gave us the opportunity to form crystal contacts, but we had no idea what this thing did. And that was the reason that we called Elena on one afternoon in New York, and she was a little bit shocked. There was some complications there because we ended up within a phone call, first acknowledging the fact that we were working on essentially the same enzyme in different groups. About a couple of days later, what became as a hostile competition became a neutral competition, and it turned on to a friendly competition where we co-published in Nature Communications in 2015 the 2 structures, and this then led to a fantastic collaboration. So our initial work, our work and the work and the let's call it the competing work of Elena, showed essentially could identify a role for each one of these 8 conserved residues. So we could understand why the signature sequence of this family of enzyme, what every single amino acid was doing in the mechanism of reaction. But a detail which is not to be neglected, this subfamily of enzyme here didn't use a lipidic substrate as a substrate. So it was in a way marginal in enlightening us of how the glycerol phospholipid biosynthesis was going on. So then we went from this friendly competition with Elena to a tight collaboration which lasts up to date, in which we did some protein engineering. We took the domain of one, put it on the other, and we were able to obtain crystals of a CDP alcohol phosphotransferase that does indeed carry the lipidic substrate in the form of CDP diacyl glycerol. We were able to use lipid cubic phase crystallization to determine the structure with and without the lipidic substrate itself. And we could see how the diacyl glycerol, shown there in purple, binds to the side of the enzyme and slots the nucleotide, the CDP, straight into its active site. Some elegant structure-based mutagenesis performed by Elena's group led to our first joint publication, which was a 2016 Nature Communications, a paper that as of today I'm extremely proud of, and which set the basis of our future collaborative effort. So this was us, but the problem in the field, in the broader field of membrane protein crystallization, crystallography, remained. We still had to deal, even by going down all the steps of the process and identifying those that could really stand a chance of yielding crystals, we still had those abysmal statistics in terms of obtaining initial crystals. However, we had spent about 20 years optimizing the biochemistry to get this point, to make the best possible sample. So when in 2013, the group of David Julius at UCSF showed that with the advent of direct detectors onto electron microscopes and an advancement both in software and in hardware, in one moment we didn't need to create crystals anymore. We could image the protein directly in a very thin film of vitrified glass by electron microscopy. That was a eureka moment for the field. We'd spent the 2 decades preparing the sample. Now we could take that same sample and we didn't need to crystallize. And the explosion of the field of membrane protein structural biology from this advent of David Julius in 2013 has been outstanding, to the extent that if you look at the statistics over the years, not surprising, this is slightly outdated, the number of structures solved by single particle cryo-electron microscopy has outperformed those by X-ray crystallography. And the applications going forward in present and in future of these techniques are humongous. We've heard in the first lecture of the idea of taking the single particle approach and tweaking it to go into the cell in more physiological conditions. And tomography, we've heard about the capability of studying less abundant, because the amount of sample you need is less than for a crystallization experiment, and progressively more complex system. As a lab, it opened us 2 possibilities. One is the progressive study of smaller and smaller systems as the technique has progressed. It's a microscope. You've got to look. Size matters, obviously, in the sense that the larger the object, the easier it is to see. The smaller the object, the more difficult it is. But the technique has improved, as is shown in the left here, of progressively smaller proteins be tractable by the technique. And for the membrane protein field, it has allowed us to develop techniques to image the membrane protein in a close to native environment of a reconstituted nanodisk, i.e. in a lipidic environment. And my lab has, in a way, pioneered some of these 2 highlighted aspects, and we've been able to show in our 2 themes, one being the interaction of the membrane protein and the membrane for the transport of lipidic substrates. We have shown over the years how, for example, like vitamin A, the hydrophobic retinol, essential for the visual cycle, is taken up into the eye through its receptor STRA6. Or we've more recently shown how the omega-3 fatty acids, those abundant in fish, get taken up across the blood-brain barrier by a specific transporter called MFSD2A. And we've shown in the Wnt, in the classic Wnt signaling pathway, how the attachment of the lipid to the Wnt is essential for their short-range transport and signaling by the carrier Wintless. And we've also used a combination of X-ray crystallography and single particle cryo-EM to target a second interest of the lab, which is to understand the molecular mechanisms of drug disease, of drug resistance. We've shown in an early work, first by X-rays and more recently by unpublished by cryo-EM, how this enzyme here modifies the charges on the lipid A component of the outer membrane of gram-negative bacteria, preventing the efficacy of polymyxin, last resort polymyxin antibiotics. And we've shown how this nasty molecule there, or PF-CRTs, chloroquine-resistant transporter, is at the basis for the historic failure of chloroquine as an all 4-aminoquinolines as antimalarial drugs. But what I want to tell you today is to focus on a more recent story, which essentially combines all these interests of the lab, these two interests of the lab. And let's just take, again, a little step back and look at these nasty bugs called gram-negative bacteria that we all know about. So what makes them particularly nasty is the fact that they have two membranes. And the outer membrane, which is the first point of contact with the outer, with the host, with the host victim, is constituted in its outer layer by what's called a mixture of sugars and lipids called lipopolysaccharide. Lipopolysaccharide, or LPS, shown there on the right, is constituted by a lipidic component which is called lipid A, which attaches itself into the outer membrane. A more conserved oligosaccharide component, which is called the core oligosaccharide, and an extremely variable repeat of 3 to 6 sugar units called the O antigen. Now, the O antigen is the first point of contact with the host. And so modification in O antigen can modulate both toxicity, immunogenicity, and can lead to immune evasion. So it's an extremely important mediator, the O antigen component of the LPS, of how the host, us, interacts with the gram-negative bacteria. And so it's a powerful tool for the gram-negative bacteria, a powerful tool in defense against the attacks of the immune system of the host. So the way this is made in two components. It's a complex enzymatic reaction made in two components in the bacteria. On the right, shown here, is the lipid A part. Lipid A and core oligosaccharide are made in a conserved, what's called the MSPA pathway. The O antigen, which is extremely variable, even within the same bacterial species, different strains can have different O antigen components, can be made in a variety of enzymatic pathways, both ATP and ATP dependent and ATP independent. And it's in a cascade of enzymes that assemble this repeat of sugars. And then they've got to be all come together. And this happens in the inner membrane of the gram-negative. And the reaction is catalyzed by a single enzyme called WAAL, also known as the O antigen ligase. O antigen ligase because it ligates the O antigen component to the core oligosaccharide of lipid A. It's a sugar, it's sugars with sugars, so it's a glycosyl transferase reaction, schematized here on the left and shown on the right as a reaction in which the O antigen, this highly variable polysaccharide, is linked to a lipidic carrier, which is called undecaprenol pyrophosphate. So these are 11 prenyl units, 2 phosphates, and O antigen. This O antigen is enzymatically taken off the 2 phosphates and put and onto the core oligosaccharide of lipid A to form the mature LPS, which is then transferred through what's called the LPT pathway to the outer membrane of the gram-negative bacteria. So we became interested in WL because it was the missing link in the biosynthesis of LPS. Let's see if the computer agrees with our interests. Yeah, so we did what we had learned to do, and we screened different orthologs of this enzyme across different species of gram-negative bacteria, and we could see some expressed, some disexpressed, as shown in these Comassi-stained gels here. We chose one that expressed the best and was most stable in detergent. And so they're not, hold on a second. Okay, so this was the WAL from a gram-negative bacteria called Cupraviridis metallidurans. We had to go into Cupraviridis to actually make sure that what we thought was a WAL was a WAL, so we had to genetically modify that. We tried to crystallize it to no avail, and then we put this thing, we reconstituted in lipid-filled nanodiscs, and we put it on a microscope, and the results were pretty tragic. All we could see were discs. We could see no structural features from the electron microscope. This was in part, two things was due to. One, that this thing sits in the membrane and doesn't have much outside the membrane that you can see. Having things outside the membrane is important in single particle analysis because you need to align and one particle with the other. But most importantly, when the limit of the technique is about 100 kilodalton, we were dealing with something which was 44. So it was maybe an experiment we shouldn't have even done. So we turned to a technique that we've used in several occasions with success, is in which we use, through a collaboration with the group of Tony Kosikov at the University of Chicago, we use a synthetic antigen fragment, in this case FAB library, which has been engineered, one, to provide rigidity at the hinge region on the left, and two, it's a synthetic library to provide as much variability as you can in the region of this antibody fragment that binds the antigen. And the way this is done, it's done by a technique called phage display. So the coding sequence for the antibody is attached to a phage so that you can go from the phenotype to the genotype quite readily. And without going too much into the details, the protein reconstituted in a biotinylated nanodisk, shown there in the scheme, is used as a bait to fish, to essentially fish for binders against your protein. And then the empty nanodisk, those that will bind to the nanodisk, but not your protein, are discarded away by abundant washing with empty nanodisk and by rounds of enrichment. It's a tedious procedure, but it's a procedure that works. And it worked extremely well in these circumstances, and it had two effects. One, it increased the size from the 44 kilodalton to about 94, because an FAB is 50 kilodalton. And that put the project into what is tractable by single particle cryo-EM. And two, it added features. The FAB added structural features outside the nanodisk that we could use to align one particle from the other. So if we look at the initial, so we were able to obtain 6 binders against this. We used some biochemistry shown here by size exclusion chromatography to reconstitute the complex, shown in chromatography in the center and on an SDS page on the right. And the initial results by single particle cryo-EM were great. You could immediately see that the micrographs look good, even on a non-expert eyes. And if you look carefully on what are called 2D class average, you can start to see these little lines inside the nanodisk, which will end up being the representation of the transmembrane regions of the enzyme itself. So a few months of processing of the data later, we could obtain what's called a density map of the antibody enzyme complex, shown here. And this is the final map at about 3 angstrom resolution, in which the protein, the transmembrane enzyme, is shown in rainbow colors, and the antibody binding on top is shown in gray. From the density map, we could build the structure of the enzyme, which is shown here, and it shows a rather characteristic fold for membrane-bound glycosyl transferases, with one interesting exception. So it's a 12 transmembrane fold. It has a top periplasmic domain and an interesting helix highlighted there as transmembrane 9, which kind of sits there outside of the membrane, in a way minding its own business. So we went a little bit further into an analysis, and we looked at conservation, and we looked at electrostatic. Conservation on the top showed that the hotspot of conservation was in a patch which happens to be positively charged. You see the analogy with the bottom panel, and on a crest above, on the periplasmic side of the enzyme. So we then looked for cavities, and we looked where this patch corresponds to. And if we looked where the yellow cavity is, which is where the transmembrane 9 and the cluster, it turns out, of arginines dictates, of extremely conserved arginine, dictates the positive nature of that region, we could see density, clear density in that cavity, in the green cavity, that we could interpret as a part of the undecaprenal pyrophosphate. So we could essentially see two of the prenal units, there are 11 of them in the carrier, and the two phosphates, which is, we interpreted as being a product of the reaction. We confirmed, and the two phosphates are coordinated by these positive charges from the arginines. We used genetics coupled to functional analysis to highlight the fact that the arginines are two of the four arginines are essential for the enzyme to work. So, and then we took it a step further. We had to confirm that what we thought was undecaprenal pyrophosphate was indeed that. We used pretty crude methods of thin layer chromatography shown here. And then we turned to molecular dynamics simulation, where we took the structural biology model and we brought in computational approaches to show that if we mapped the entire substrate or the entire product on where we could see only 2 of the 11 prenal units, and we let this flow in molecular dynamics simulations, it actually stayed where we saw it, and that the part that we didn't see, shown there in red, was actually the most mobile part of the undecaprenal pyrophosphate, making sense of our experimental observation. We looked back at the micrographs, and we noticed that we had another class of, in the same collection, we had another class of proteins in which that density that corresponds to the product wasn't there. So in one collection, it turns out, we had both a product bound and an apo form of the enzyme. The two are similar, are extremely similar, if not of some movement, not surprising, of the arginine themselves. But if we take the two models, one of the apo and one of the product, and we subject them again to a round of molecular dynamics simulations, we could note that while the product-bound form was extremely stable, the apo form showed a movement, and it showed actually two states shown on the right of transmembrane 9, of this transmembrane helix that sits outside, allowing us to make the hypothesis that the movement of this transmembrane is actually how the substrate comes in for catalysis to occur. Now, the enzyme takes an undecaprenal pyrophosphate linked to antigen, but it also needs to accommodate this massive lipid A with the core oligosaccharide. So where can that sit? And that became a little bit more controversial, and we went a bit speculative and turned to the other side of the molecule and highlighted in the blue cavity, or cavity 2. The view from that side is shown on the bottom, colored by electrostatic, hydrophobicity, and conservation. Now, it's tricky here because lipid A varies between species. It's not always conserved. There are variations. There are variations in size, in length of the A cell chain, but this had not decisive evidence allowed us to postulate that that could be, on that side of the molecule, the lipid A binding site. In support of this, what did we have? We observed in our maps, calculated our experimental maps, that the membrane shown on the left deforms on that side of the molecule, consistent with the length of the acyl chains of lipid A, which are shorter than the typical acyl chain that constitutes a biological membrane. We could do the same experiment by molecular dynamics simulations, i.e. computationally, and we could show that this would led to the same deformation of the membrane due to two transmembrane helices of the enzymes that are shorter than the typical transmembrane helix. We could then use again computational approach and simulate lipid A binding, and there was a stable adduct of enzyme with lipid A. And more interestingly, if we docked the lipid A on that side of the molecule, and we had the core oligosaccharide span across the top of the enzyme, we could see that the lengths, the theoretical lengths shown on the right, we need to span 25 angstroms from the hydrophobic tails to the tip where catalysis occurs, and the length between the observed product and this binding site corresponds, would make sense. So taken together from these data, we could speculate and we could hypothesize and make a model of how this enzyme works, in which the O antigen linked to undecaprenal pyrophosphate comes in with a movement of this atypical transmembrane 9. The lipid A binds on the other side, and catalysis happens on the top of the periplasmic side of the molecule to create and then release the mature LPS, which is then transferred to the outer membrane by the LPT transport system. And we could take this one last step further and ask the question, well, how does catalysis occurs? And of all the residues across all WALs, 2 of the 4 arginines are always conserved, as is a key histidine showed there in green, which sits on top of the arginine in between what we hypothesize as being the lipid A binding site and the undecaprenal pyrophosphate O antigen sites. So this allowed us to speculate on the mechanism of catalysis in which the histidine abstracts a proton from a sugar and leads to a nucleophilic attack, and to make a first hypothesis of how this glycosyl transferase works. Glycosyl transferase that we discover is actually similar in fold. It's a broadly related fold of what are called the glycosyl transferases type C, because they type C, the membrane-bound glycosyl transferase, but it shows particular analogy or structural homology to another enzyme that is essential for making the polypeptide, the polyglycan of the peptidoglycan in gram-negative bacteria, which is a protective layer that sits under the LPS and above the inner membrane, and it's an enzyme called ROD-A. You can see the similarities in this extra helix, this 9 that sits out, let's say, in the middle of nowhere. Both enzymes, the enzymes catalyze a completely different reaction, but they both, one of the substrates is undecaprenal pyrophosphate link. And if we look more broadly at these glycosyl transferases, and we look at others that use either undecaprenal phosphate or they're undecaprenal with 2-phosphate, you can see that they all have a periplasmic helix shown here in blue or shown here in here, where one arginine sits there and coordinates the phosphate of what's called the donor substrate, suggesting an evolutionary conserved mechanism for the entire family of enzymes. And at this point, I think I have come to an end of what I wanted to tell you, and I'll just summarize this part here that was published in Nature last year, is that we use single particle cryo-EM to determine the structure of the O antigen ligase. It's a 44 kilodalton enzyme. We had to go up the wall a bit and tweak the antibody part and the nanodisk in order to be able to obtain this. We hit lucky, and in one collection we had a product bound and apo form of the enzyme. We could, in the product bound, we could see that a set of arginines that tend to hang out with each other, having a good time with the undecaprenyl pyrophosphate. We were more speculative on the lipid A binding site, and that led to mechanistic hypothesis on how this enzyme work. Enzyme that has a conserved fold, very conserved and conserved in some features with the broader family of membrane-bound glycosyl transferases. Last but not least for this part, I'm a strong believer of collaborative science, and it actually happens that just by chance on this project here, there aren't any Portuguese collaborators because I have had a great time collaborating with Portuguese groups. And I'd like to mention the hero, my former postdoc, soon to be senior scientist at Schrödinger in Boston, Khurram Ashraf. I'd like to mention Ollie Clark, who is our local single particle cryo-EM expert. Tony Kosiakov in Chicago for the work in generating the antibody fragments. The bacteriology and the functional assays were performed in close collaborator, Stephen Trent's lab at the University of Georgia, and Anne-Catherine Uhlemann at Columbia helped us with the genetics of showing in this Cupra viridis metallidurans that this was actually a WL. And last but not least, I have Phil Stansfeld at University of Warwick, again, a collaborator for the molecular dynamics simulations, and you need microscopes. So I'm extremely grateful to both the New York Structural Biology Center and the Columbia University cryo-EM facility. Thank you.
Thank you, Filippo, for a beautiful story. And now I'll ask Professor Maria Salomé Paz to close this wonderful session.
It's my pleasure to close this so interesting and so strong session. And I hope that this is an opportunity that will be able to be repeated, because it was very, very, very useful for all of us, with so good results and so well presented. So thank you all. And now I would like to invite you to take a small tea outside or inside. And if you have time, we can, instead of leaving the academy from the door you have entered in, you can go out from another way, passing through the building, in order to have an idea of what is there.